My name is Lydia Morley, and I'm at Texas A&M University, and today I'm going to talk to you guys about the climatic and phylogeographic correlates of hybridization in vascular plants, globally and regionally. Okay. So the last decade or so has obviously seen a transformation in our understanding of hybridization. Evidence has continued to pile up for its importance and prevalence in evolutionary histories across the tree of life. And this is perhaps especially true and time-tested for plants. The importance and prevalence of hybridization across the tree of life is, in and of itself, something important to investigate. But this revelation should also motivate us to rethink the kinds of questions we ask about evolution itself. We often ask, why are lineages where they are? Or we consider the interactions between speciation and extinction as causal means to an evolutionary end, which is extant biodiversity. But sometimes we want to ask, why do things have the evolutionary histories that they do? In other words, we should investigate what drives the shape of evolution itself. We should care about why and how lineages come to have the particular evolutionary histories that they do. And critically, investigating this shape of evolution requires us to consider not just speciation and extinction, but also gene flow, or hybridization. In essence, we aim to discover what things drive this push and pull between divergence in gene flow over time, and whether they leave observable patterns in extant biodiversity. In this way, we hope to show that hybridization is not just like an aberrant, albeit common phenomenon, but rather, like speciation and extinction, is a predictable and essential feature of lineage evolution driven by phylogenetic, climatic, biogeographic, and historical factors. So throughout this talk, we'll be discussing exactly what those factors are and other fun and exciting things. So recent investigations into the climatic causes of hybridization, such as those recently published by Folk et al. In that group, have given weight to Anderson's disturbance hypothesis. And basically, this hypothesis states that lineages are liable to hybridize when the spatial or ecological status quo is disrupted. The causes of disturbance might be anthropogenic, glaciation, the formation of mountain ranges, anything. What matters is that when adaptive landscapes change, post-speciation gene flow happens. The way we see it, disturbances facilitate hybridization in 4 ways. First, species that were previously separated may come into secondary contact. Second, populations that were previously in contact may become allopatrically separated, leading to divergence that potentiates gene flow upon future secondary contact. When climatic and geographic landscapes are continuously dynamic, the former and the latter occur cyclically, leading to overall increased rates of hybridization and hybrid survival. Third, dynamic ecological and geographic spaces can open up previously unavailable niche space for hybrid offspring to survive away from either of its parents, and perhaps for adaptive introgression to occur successfully. Finally, species that survive over time in continuously dynamic landscapes tend to be more migratory and more generalized than those that don't. I have some citations for that if you're worried about that big bad statement, but seems like, at least for plants, it's shown to be pretty reasonable. The lack of ecological specialization, coupled with an ability to move around when landscapes change, means that those lineages might intrinsically have lower barriers to gene flow, and hybrids of 2 generalists might be more apt to survive than hybrids of 2 specialists. So in general, climate dynamism and landscape disturbance facilitate hybridization by providing increased hybridization opportunity, increased survival opportunity for those hybrid offspring, and by creating an evolutionary landscape in which lineages that intrinsically have decreased barriers to gene flow are potentially more likely to survive. So all that being said, we feel like we should expect to see greater hybrid content in temperate regions where both hybrid opportunity and hybrid survival have been historically maximized. And we expect to see variables associated with this disturbance through time coming up as important for determining variation in hybrid content both globally and regionally. So just as a first look, we do see this. Great. So here we measured hybrid content as a log proportion of total species richness that is comprised by named hybrids or nothospecies from the World Checklist of Vascular Plants. This is actually super conservative measure of hybrids in an area, but also considering only nothospecies gives us 2 advantages. So first, we can reasonably assume that these gene flow events are happening right now or have happened relatively recently. Given that many hybrid species and hybrid zones are ephemeral in macroevolutionary time, the current global hybrid content should be reflective of relatively recent bouts of climate dynamism and gene flow events. Second, with all this data, it was basically just the easiest thing that I could do. So, using this as a jumping off point, this project uses random forest modeling for explaining the occurrence patterns of hybrids to test the hypothesis, again, that historically dynamic climatic and phylogeographic regimes positively impact. I just jumped 6 slides. Okay. Positively impact hybrid content. Okay, so to assess the correlates of hybrid content, we gathered over 370 million species occurrence records for all tracheophytes from GBIF. We cleaned them. We cross-referenced them with our POWO data, Plants of the World Online, and bin them in equal area grid cells. We then use those grid cells to collate sets of current and historical climatic records, along with ecological and geographic variables such as fire frequency and elevation. Using the massive chronogram, there we go, generated by Smith and Brown, we then calculated phylogenetic community indices for each cell, such as NRI, mean family age, degree of congener overlap, and mean DR statistic scores for a given cell. So at this point, it's important to note that all of our tested variables only indirectly affect observed hybrid content via some path through hybrid opportunity/frequency or hybrid survival, or both. At the same time, we have what's known as zero-inflated data. That is, for most areas of the world, there are few, if any, hybrids at all. So we additionally implemented a 2-stage modeling process, asking first, what factors correspond to hybrid occurrence in general? And second, what factors correspond to differences in hybrid abundance where hybrids occur? So we started with a lot of variables, and in order to narrow these down, we first separated them into 3 categories, those related to temperature, precipitation, and those related to phylogenetic community structure. We performed random forest analyses and GLMs on each of our partitioned data sets, and then combined the important variables retrieved from each into a composite data set. We ran global analyses on the composite data set, and then we repeated the whole process for tropical, temperate, and continental regions. We did this 4 times, modeling hybrid content, hybrid occurrence, hybrid abundance, and total species richness. In addition, we included GDP and HDI as predictors in all of our analyses to account for the possibility of acquisition or taxonomic naming bias in areas that are more heavily funded and studied. So I'm just going to show you guys an example of how things change when we implement single versus 2-stage modeling. So first, we can clearly see that the important variables determining total species richness are different than those that are important for hybrid content. In other words, increased species richness doesn't necessarily always correspond to increased hybrid content. This pattern is repeated across regions. For hybrid content, major drivers are human development index, temperature stability, and mean community-wide DR statistic scores. If we look at regional differences in the correlates of hybrid abundance, we can see some slightly different patterns, and we also see this global pattern kind of breaking down in specific areas. So in this heat map, colors correspond to the directionality of a relationship, while opacity corresponds to its importance. We can see that many regions exhibit the same relationships with most variables, though their importance is variable among them. Global and temperate regions exhibit stronger relationships with temperature and precipitation stability, for example, while tropical regions exhibit stronger relationships with species richness and mean nearest taxon distance. This points to the importance of local, historical, and climatic regimes in driving observed patterns of hybrid abundance. At the same time, we have to note that both HDI and GDP were moderately important across all models, indicating that there is some bias happening here. Maybe we can say, however, that at least the bias is uniform across all regions, and so equally affecting all of them equally. But right now we're working on mixed effects random forest modeling to better understand the kind of joint and individual impact of these variables, including those that represent bias. Okay. I forgot that I had boxes on all of the variables, but okay. So each tip on this tree is a genus, and each color column represents an area. Gray cells are places where that genus does not co-occur. Each member of this tree contains at least one hybrid somewhere. Globally, we found no phylogenetic signal in hybrid content, and we can see that genera with many hybrids in some regions have few or no hybrids in other regions. This means that we can't consider hybrid propensity to be driven by phylogeny alone, but rather, again, that it's contingent on a combination of phylogenetic, geographic, and historical factors. Okay, so what we found so far. Obviously, drivers of hybrid content are different from the drivers of species richness. Across regions, species richness is an important variable for determining hybrid occurrence, but not always for hybrid abundance. So here it's not the case that we're just always seeing more hybrids where there are more species, but it might be the case that we have a higher chance of seeing any hybrid at all when there are more species. Other important drivers of hybrid abundance are overall temperature stability and extreme climatic conditions, along with species-specific diversification rates and HDI. And we also found that lineages don't express uniform hybrid propensity across their ranges, that it's variable and likely dependent on these other factors that we tested. Okay, so key takeaways are things that I hope that you think and that this presentation convinced you to maybe think a little bit more strongly. So gene flow or hybridization is a predictable and measurable feature of lineage evolution driven by historical, biogeographic, and phylogenetic factors. I've said that like 6 times, so hopefully you agree. Investigations into global patterns of biodiversity should focus not just on outcomes of evolution, but on the shape of evolutionary relationships themselves to better understand how these processes are happening through time and space and why. And of course, better understanding these processes are invaluable for predicting how lineages will respond to future change and also understanding how they came to be in the first place. Okay. I would like to thank all of my lab mates and other grad student colleagues who have watched like thousands of iterations of this talk over many, many, many months. So thanks to them, and also my cats and funding. Okay. Three minutes for questions.
How about hybrids being more abundant where they are studied? Because they're not studied all throughout the world equally?
Mm-hmm. Yeah. So I did talk about that. So she's asking about hybrids being more abundant in areas where there is taxonomic or acquisition naming bias. And we did include GDP and HDI as predictors to see if, as representatives of bias, that those can give us some kind of thing. And by implementing mixed effects models, we can basically like partial out the effect of that bias and just look at how the other variables outside of that naming or acquisition bias might contribute. We also are thinking about a different kind of measure of naming bias. And naming bias is just things being more abundant in areas that they're studied, right? Like you find more hybrids in areas where there are more people studying things, right? Yeah.
That's so biased in
Mm-hmm.
species-rich countries.
Mm-hmm. Yeah. And I mean, that's a problem with global analyses of species richness, just like in general, right? And so also, I mean, one of the things that we did is we structured these things. We restricted them to specific areas as well, like we did the regional analyses. And so the global analyses are probably pretty biased towards temperate regions or wherever, right? But temperate regions themselves are perhaps not biased to temperate regions. And the signal from temperate regions is potentially less biasing of the data when you exclude it from analyses and look only at kind of like tropical regions together or something like that. So, yeah.
My name is Trevor Faske. I'm a research ecologist at the United States Geological Survey in Flagstaff, Arizona. And what most of my research group does is we use genomic data and landscape genomic approaches to inform conservation and management of plants in the western U.S. And through that, we end up analyzing a lot of plant genetic data sets, and we kind of see these replicate patterns across a lot of them in that region. And first off, plants are weird. That's the first pattern, right? They hybridize like crazy. They're mixed ploidy. They do all this weird stuff. And secondly, they also share these kind of commonalities with the glacial cycles that happen in that region, and also the environmental and topographic heterogeneity. And so I'm going to highlight some of those patterns that we've seen across a few different taxa. And I'm going to do acknowledgements first. So this is like a big science project from everyone in my academic family tree, which has been really fun to do, and then a bunch of funding sources. So evolutionary history and secondary contact. So in North America, there's been around 11 glacial cycles over the last 2.6 million years. And with this, genetic variation across this landscape is partitioned because of these glacial cycles. So once they happen, so there could be allopatric isolation that diverge species, and then they have different niche divergences and occupy different environments according to these glacial oscillations. And then after they recede, this is just an example of some expected post-glacial expansion routes. They come into secondary contact and hybridize in these region of intermediate environments. And so we know that hybridization happens at these transitional environments. So this is just an example with Encelia, this desert plant that has a dune species and a desert species, and it kind of hybridizes at these intermediate ecotone environments. And they also occupy unique environmental space compared to the parental taxa. And in the Western US, we have an abundance of these transitional environments. So this figure that you see here is just an ecoregion map. So basically it's just a climate matching map. And you can see there's lots of topographic diversity and also regions where these transitional zones can occur. So if we look at a classic hybrid zone, this is an example in a pine species where you have this like northern species and a southern species, and then there's a hybrid zone right in the middle at this geographic line, a single point of contact. In a mosaic hybrid zone, that's not what you see. A mosaic hybrid zone is where the reproductive isolation is due to some ecological or environmental variables and rather than a single geographic line. And so some of the previous work we've done in rabbit brush kind of shows this. So the gray is one subspecies, the green is another subspecies, and the yellows are the hybrids. And you can kind of see hybrids pop up all over the place. The greens and the grays, they kind of co-occur sympatrically across their range. And then the hybridization is predicted by these ecological environmental factors. So our objectives for this project is we're going to try to ask, do glacial cycles lead to convergent patterns across plant taxa in the West? What ecological factors drive this hybridization, and can we predict it to some degree? And what are the future and current ecological ramifications of this mosaic hybridization? And so the three species I'm going to show you today are rubber rabbitbrush, big sagebrush, and Findler's globe mallow. And so all of these species are of restoration concern in Western North America, but they have some key differences. Rubber rabbitbrush and big sagebrush are shrubs, foundational shrubs that occupy kind of vast geographic ranges around the entire West. Big sagebrush has been of conservation concern for a long time. Globe mallow is also this like really messy species complex. It's a forb. It's a known syngamian. It kind of disturbs colonizes disturbed areas. And so I'm going to show you a figure for all three of these coming up. So it might be best just pick your favorite, and then I'll explain that one. That's going to be the easiest. So with these are our structure plots for each of these, and this is how we decided between the parental ancestry on the left and right, and then with the hybrids in the middle. And so the hybrids are yellow in each of them. And you can kind of see that the parental taxa kind of co-occur sympatrically across their range, where the hybrids kind of pop up all over the place. And so throughout this talk, I'm going to talk about some convergent patterns that we're seeing in these very, very different taxa. They have a lot of differences, but there's a lot of similarities that, within them as well. So first, do glacial cycles lead to convergent patterns? So how we attempted to do this was we did a demographic inference of the divergence in the parentals between the parental species. And we used some like common models in FastSimCol to do this. And overwhelmingly, all of them showed this model or predicted this likelihood of a secondary contact model, which is just where they diverged in allopatry some long time ago, and then time passed, and then they started hybridizing or having some gene flow between those two species. And so if we look at the timing of divergence, that varies drastically across all three of these species from 700,000 to 80,000 years ago. But if we look at the timing of secondary contact, it's all within the last one or two recent glacial cycles. So we can look at what ecological factors are potentially driving this hybridization, and can we predict it to some degree? And instead of trying to show you this for all 3 species because it gets really overwhelming, I'm just going to show you for big sagebrush. And big sagebrush is a really common restoration species or conservation species in Western North America. Millions of acres of these shrubland communities have been converted to these exotic grasslands due to fire and exotic grasses being introduced and all the bad things. It's also a really important habitat for this crazy looking chicken that people care about. And so, yeah, that's why they've been doing a lot of conservation on this species for a while now. And it has 3 main there's polyploids in here that I completely ignored. And if you're interested in mixed ploidy population genetics, we can talk about that some other time. But so we're just looking at the diploids here that segregate environmentally across this elevational gradient that you see here. So there's this Phasiana that's at higher elevations, that's colder and wetter, and then this basin sagebrush that's down in the basin. And so they segregate elevationally, and they're known to occur sympatrically across the range, besides for they're segregated environmentally. And so if we look at a PCA of our genetic data, we see exactly what we expect, where the 2 parental taxa are diverged, with hybrids occurring genetically intermediate to this. And we can look at how environment correlates to that. So this is a redundancy analysis or a genetic environment association. These are commonly used to they do kind of a bad job of identifying loci that are associated with environmental variables, even though we all do that a lot, but they do a really good job of explaining the spatial genetic structuring of a species across the landscape. And so the way you can read these is the individuals in red, the Phasiana, are more heavily associated with increased precipitation seasonality, increased elevation, which we know. And the other way, the blue ones are associated with climatic water deficit. And that's in a multivariate framework. We can also look at the same thing in a univariate framework. This is a beta regression, and we see that hybrids are indeed environmentally intermediate to the other 2. We can also bend it, and we see the same thing across these 2 major environmental axes. I'm not going to show the rest of the other species, but they show very similar patterns. There's some overlap in the environmental variables that are correlated with these 2, but overall it just shows that environmental space is explaining the niche divergence between these 2, and hybrids are indeed intermediate, even though they co-occur sympatrically across their range. Next, we can ask, can environment alone predict the occurrence of either of the parentals or the hybrids? And it can, to a surprisingly high degree. So what this is a machine learning model tested and trained just using environmental data and ancestry coefficients. And so doing that, we can predict ancestry or the occurrence of one of these parentals, or either these parentals or the hybrids, 88% of the time. And even in Phasiana, we can do it 97% of the time. If we look at all 3 species, we see the same thing, right? To a very high degree, we can predict the occurrence across the landscape just using environmental data of one of the parental taxa and also the hybrids, around 88% of the time, and some of them almost 100% of the time. And so with this, because we're able to kind of predict the occurrence of ancestry across the landscape or the occurrence of the parental taxa, we can ask, what are the current and future ecological ramifications of this mosaic hybridization? And so what we did was, now that we have these predictive models and we test and train them on our data, we can project it across the entire range of these species. And we can calculate the amount of area that is occupied by the hybrids. And so this is for all 3 species. It looks really messy, but the hybrids are in yellow. Again, and we can calculate the amount of hybrid area. So in rabbitbrush and globe mallow, it's around 20 to 25%. Have a take a drink of water. And within big sagebrush, it's only 0.4%, but that's because if you zoom, you can't really see the yellow, but if you zoom in really close, it's because it's at that really sharp elevational gradient, and if you looked at any mountain range in Nevada, it's in there in the middle. And so with that, hybrids are occupying unique niche space between their parental taxa and potentially providing some sort of ecological importance there, or at least creating connectivity across the range, because it's increasing niche space that the entire taxa is occupying across the range. And we can ask, what do we expect under future climate change scenarios? And we can project these into the future. And under each of our climate change scenarios, I'm just showing one of them, we did a bunch of them, you can see that overall expected hybrid area is expected to increase over time, while total area is expected to decrease over time. And especially, usually one of the subspecies or lineage is expected to drastically decrease. And we see the same thing in all three species. In sagebrush, it's expected to go up by 500% almost, while overall area is expected to decrease. And same thing within globe mallow. So overall conclusions, I had a different conclusion side, and then I was going to have a big conclusion side, but we just have a big one now. So glacial cycles and the topographic complexity of Western United States is probably driving a lot of these patterns, and it's probably occurring at a way higher rate than we're actually seeing in plants, or that's been documented so far. I think there's a lot of hybridization in plants of restoration concern, or plants in general in the Western U.S., and this is a likely pattern that we'll see in a lot of things if we go out there and look for it, mosaic hybridization across there. Also, hybridization is predictable to some degree, and it increases the overall range of taxa, and increases connectivity across the landscape. And so I work a lot with land managers in trying to move things across the landscape, and they're going to do it anyways, and we try to inform them. So we need to start potentially considering hybrids and what to do with them when we start moving things across the landscape. And lastly, this is something I'm personally really interested in, is can we predict ancestry across the landscape? So a lot of this conference has been about prediction, or at least a lot of the talks I've been to, and we can do kind of a bad job at it at certain levels, but maybe at greater scales like population structure or ancestry or something, we might be able to predict that across the landscape, and that could be used really heavily in management when you're considering moving things across the landscape. And that could be something that's really simple to give to land managers for them to kind of use to make those decisions. Yeah, so that's something I'm interested in working on. If anybody wants to talk about that, I'd be happy to take some advice. And with that, I'm done. This is my dog in front of a giant rabbit brush. So, thanks.
Quick question. So the Western U.S. is obviously, as you mentioned, really topographically dynamic. Do you think that your conclusions might hold true for other regions of the world? Assuming glaciation history.
Yeah, probably. I mean, I don't know. I'm kind of ignorant to other regions of the world. I know glaciation events in Europe, right, are east to west. I'm sure you can kind of see this pattern, but I haven't looked too deeply into that research over there. But you actually don't really see mosaic hybridization papers or things in general on plants, and so it's not really heavily. Yeah.
I'd like to talk to you about that actually.
Okay. Cool. Thank you.
I'll show you the email. Thanks.
So hello everyone, thank you so much for being in my talk. My name is Sara Carvalho Madrigal. I recently got my master's in biological science at Universidad CES in Colombia, and I recently a research assistant at Arizona State University. So today I'm going to share with you the main results about my thesis research, which is about the role of introgressive hybridization in peripheral, founding, and expanding populations of wax palms, genus Ceroxylon. So before diving into the context, I would like to start by talking about this extraordinary and interesting group of palms, the wax palms of genus Ceroxylon. And this video was taken on the biggest wax palm forest in Tochecito Valley in Colombia. So this genus is formed by 13 species, which are all endemic to the Andes Cordilleras, and they are distributed from northern Venezuela to southern Bolivia. And something interesting about these palms is they're extremely tall palms, reaching 60 meters of height. And you can see this arrow, it's pointing a person standing next to the pond, so just you can see the scale of how tall are these ponds. And they hold in the records of the tallest pond in the world. Also, they grow at the highest elevation in mountains, and they live in environments that are often moist, either by rain, mist, or low clouds. And as you can see in this picture, it's the most common environment when these palms live, which is always covered by clouds. So 5 of the species that are adapted to the highest elevation range, between 1,900 and 3,500 meters above sea level, form a monophyletic group, but these 3 species show inconsistent relationships in recent phylogenies and were considered a species complex, the Kindiwense complex. Also, Carreño and others studied the reproductive biology of these species respective to these 3 species, Ceroxylon ventricosum, Ceroxylon parvifrons, and Ceroxylon vogelianum, which are sympatric and have a very different morphology. And they found that these species share the main pollinator, which is a sub-beetle of the species Misropus rotundula. And also they found that these species flower at different times of the year. So in this figure, each color represents one of these species. And as you can see, each species has a clear flowering peak for both staminate and piscillate inflorescence. These palms are dioecious, which it means that we have individuals females and individuals males. And also you can see that it's a slight overlap between some of the species with having like overlap flowering times. And these findings suggest that genetic exchange is possible. Indeed, the demographic history of highland groups shows that recurrent gene flow has occurred since the Pliocene. So these figures represent the most likely demographic model between parvifrons and Kindiwense complex, which are sister clades. And these sister clades diverge by isolation with migration, with heterogeneous gene flow through the genome, and by population expansion. So we thought, how is they share the pollinator? There is evidence of historic gene flow. They are sympatric in some places. The taxonomy is really consistent through all range distribution, and they stay highly morphologically different. So the main objective of our research was to explore the signs of introgression between the 5 highland species I was talking about. And we are interested in know how much and where in the genome the introgression is occurring, if we can detect signs of selection in introgress genes, and finally, how does this ties to their national history. So through hybrid capture, we sequence almost 4,000 nuclear genes coding and not coding regions. Our initial data sets consist of almost or around 2 million SNPs, and we use 166 individuals plus an ad group. So first we look for evidence of hybridization. We calculate a hybrid index, and we use Ceroxylon kindiwense and Ceroxylon parvifrons as parental populations. And we see that Ceroxylon ventricosum and Ceroxylon seriferum were the most admixed population. So in other words, these 2 taxonomically distinct species, Ceroxylon ventricosum and Ceroxylon seriferum, are hybrid forms of Ceroxylon kindiwense and Ceroxylon parvifrons, with some proportions of Ceroxylon vogelianum, which according to a structure analysis, but for limits of time I can show you here. And something interesting is that hybridization occurs at the margin of a species ranges. In this map, the pie charts that you can see at the edge or a marginal distribution of these species is the proportion of ancestry of parental populations, Xeroxylon kindiwense in blue, Xeroxylon parvifrons in purple, and the small portion of Xeroxylon ventricosum, bogelianum, and the most asthmic population are in the north in Venezuela, which correspond to the species Xeroxylon seriferum, and in the south of Ecuador and the north of Peru, which are the ventricosum populations. And just in these locations, the population densities are low. So also we use the D statistic and F4 ratio to detect the footprints of genomic integration. So the D statistic was used to detect integration, and the F4 ratio was used to estimate the integration proportion on the genome. So we evaluate 35 possible combinations of three species using this phylogeny. And only we found 14 trios of a species with significant D values. And the F4 ratio range from almost 2% to 10% between Xeroxylon parvifrons and Xeroxylon kindiwense, and notably Xeroxylon parvifrons show signals of integration with taxa of the all kindiwense complex, which is in line with our hybridization index. Then we use the FDM statistics to find potentially introgress regions or layer regions, and I'm going to show you only the results between parvifrons and kindiwense because we use the species that are widely distributed and also at the higher sister clades. But basically, we recover the same pattern for all the comparisons. So we found several peaks of integration with FDM values higher than 0.3, but only 2 regions we found that overlap with selection scenarios. And these evolutionary scenarios are based in the correlation between 3 summary statistics, DXY, nucleotide diversity, and FSCs. So for instance, we found one region in allopatry selection, which have high FSC values. You can see the pink color ones. And also the DXY was intermediate values, and the nucleotide diversity or the pi values were very low. And this can indicate that positive selection is acting in these regions. And the other region is in balancing selection, and we found lower values of FSCs, but the mean p values and DXY values were super high. So what that means? That integration regions are rarely under positive selection, and this will suggest that adaptive integration in field loci could be related to facing environmental stress, but most of the regions are under neutral or weak selection. But why this is happening, why more of the regions are under neutral selection? So at this point, we know that a species can share genes because they have inefficient reproductive barriers, because they share the main pollinator, and also some of the species have the same peaks of flowering times, and also you see that historically they share in gene flow. And we think that gene sharing itself can be advantageous because of the recreation of a common pool gene. And this is important or could be really strategic in the context of peripheral, small, and isolated populations to maintain the diversity. And just to finish and take some home message, so small and expanding populations can be the hybridization epicenters. Integration recreates a collective gene pool, which can be particularly important in small and isolated populations. We did not find many integrated regions and also under positive selection. And finally, a species can share genes occasional or sequentially, while a cell retain their morphological identity. So I would like to acknowledge to all these colleagues and collaborators, and especially to Maria Jose for all her support. In this photo, it's an individual of a backcross population hybrid, Xeroxylon soriperu, in the Sierra Nevada de Santa Marta in Colombia. And thank you so much for your listening, and welcome for any questions.
So yeah, I guess I'll just start by saying my name is Ben Stone. I'm a postdoc working with Kerry Westinger at the University of South Carolina. And in just a second here, I'll start talking to you about some cool hybrid zones that we've been working on in the genus Penstemon. Maybe. There we go. I think it's time, so yeah. All right, so the genus Penstemon is a super diverse, about 300 species genus, endemic to North America. As you can see by the photos on the screen here, a bunch of cool floral diversity in form and function, and that diversity is thought to be a product of adaptation to different primary pollinators, which in itself is thought to be a primary mechanism of speciation in this group. And in that, one of the most common transitions that we see is this adaptation to hummingbird pollinations. And so on the top, I have an example of a typical hummingbird pollinated Penstemon flower. It's going to be pink magenta, or often more red than that, sort of elongated floral tubes and a few other features, which some of you might be aware of. And then that's in contrast to a bee pollinated flower, which tends to be shorter, bluish purple in color. And this transition to hummingbird pollination has occurred many times across the Penstemon phylogeny, at least 20 independent transitions. And through some previous work by Carey, we know a fair amount about this color transition. So it turns out that in many red-flowered penstemons, they predictably have this, it's predictably associated with the defunctionalization of this anthocyanin pathway enzyme F3'5'H. So essentially, if you have a broken copy of F3'5'H, you can't produce blue pigments, and then that's predictably associated with transitions to red flowers. And so that's true for many penstemon species, many other plant species, and that also includes two of our focal species here today. We recently published a paper about this in a bee if you're interested. So we know that transitions in floral color happen pretty frequently through these loss of function mutations, but there are, I would argue, a couple open questions about this transition to hummingbird pollination syndrome. The first being, we don't really have a good sense of the genomic architecture of any of the other, I guess, loci associated with hummingbird pollination syndrome. And I would argue that more generally, maybe as botanists, we have a predilection to sort of look at pretty flowers and assume that they're sort of adapted to different pollinators. But we actually, at least in Penstemon, I would argue, in many cases, don't have a great idea of how well this pollinator specialization actually acts as a reproductive barrier. And so I think a really good system to use to study these questions are hybrid zones that form between species with different divergent pollination syndromes. And so what I'm showing you here are two of our focal species. Penstemon rupicola is a bird-pollinated species on the left, Davidsonii on the right, a bee-pollinated species. And these hybrid zones that they form they occur sort of throughout their distributions where they overlap, but they form more or less along an elevational gradient as well, where hybrids are formed at intermediate elevations. And so why study hybrid zones? I think I'll just go through this quickly because I think many of us have sort of a good sense of this, but I consider them sort of useful as natural laboratories where you can study secondary contact. You can use them to find loci associated with reproductive isolation. You can use the natural phenotypic and genetic variation in hybrid zones to do cool things like admixture mapping or GWAS. And in particular, replicate hybrid zones can be really useful to understand how consistent some of these features are of secondary contact. So basically our setup here is I've got two different hybrid zones that form between two different pairs of species that differ in their pollination syndrome, bee and bird pollination. And so we've got one site at Crater Lake in southern Oregon, and then a second site in the central Sierra Nevada. And essentially what we do is we go out in nature, collect these natural hybrids, and phenotype in the field as well. And so this is Penstemon newberrii here in the bottom, and involves a lot of photos and measurements. Oops. Well, I guess I was moving anyway. I would be remiss not to mention all of the awesome people that helped me with the fieldwork, in particular for this project. It would not be possible without these wonderful current and former members of the Wessinger Lab. So, sort of just to give you an overview of what I'm about to talk to you about. First, we don't have even really a good sense of like the structure of this hybrid zone. Like, is this an old or recent hybrid zone? And then we'll see if we can use this natural variation to find genetic loci associated with floral traits, and then maybe see if we can get at this question of whether there's actually selection against hybridization in the first place. Again, reproductive isolation is obviously not complete since hybrids are being formed in the first place. And so again, it's sort of an open question. How effective are these divergent pollination syndromes in maintaining species boundaries, I suppose. So without further ado, we'll get into some results. And I'll also just say that this project is still very much a work in progress. Yeah, I guess I'm going to mostly talk about just the Crater Lake stuff, but there will be some inclusion of the Sierras things as well. So here we've got our Crater Lake hybrid zone, and basically the takeaway from this slide is the things that look like hybrids appear to be pretty genetically admixed and sort of follow these nice gradients. So that's nice when you're studying hybridization. You have actual hybrids. And then our parent pops are more or less genetically differentiated as well. To sort of further dig into this, wanted to, I use this software called Ancestry HMM. Essentially, you feed it SNPs that are highly divergent between parent species, and then it'll assign ancestry probabilities to hybrid samples, such that it assigns probabilities to either homozygous parent 1, homozygous parent 2, or heterozygous for each SNP. And then you can sort of plot these in this interesting way on these ternary plots, because these always confuse me. I have some sort of directions here. So we would expect F1 hybrids to be sort of in the upper triangles here, almost exclusively heterozygous. And F2-like things would be somewhere in the center here. The bottom corners would be parent species-like things. And then all the stuff along the edges there would be back-crossed individuals, potentially multi-generational back-crosses. And so what we're seeing at Crater Lake is a complex hybrid zone where we've got a mixture of multi-generational hybrids, parent-like individuals, and what appear to be potentially F1 hybrids. So we've got sort of a complex, and this to me suggests that we've got an old hybrid zone, but also still active, which is cool for the trait mapping stuff, as you'll see. So I told you we collected traits. I wasn't lying. So this is a PCA of just all of our floral traits that we collected. And perhaps unsurprisingly, the major axis of differentiation here, we can split our parent species up very nicely, which is pretty obvious they look pretty different. Perhaps more interestingly, I've also plotted admixture proportion on here. So it's basically stuff that's phenotypically intermediate is also genetically very highly admixed, which is cool. And then some of the more admixed things are also sort of clustering with phenotypically with the Davidsonii-like stuff on the left. And another way to look at this is just like plotting the ancestry proportion, or the admixture proportion directly against the trait of interest. In this case, I've chosen short stamen length, but could have chosen many of our traits here. Essentially, we see this tight correlation between ancestry proportion and the traits of interest, which suggests that the traits that we're measuring are actually heritable, which is nice, and then also potentially an underlying polygenic architecture. So as I mentioned before, a cool thing about these hybrid zones is that we can leverage this natural variation to do GWAS, essentially. So we can go out and collect stuff in nature and And because there's lots of recombination happening, lots of mixing up of phenotypes in the underlying genetic stuff, we can run it through a GWAS and see if we can pick out the genes that are important for our traits of interest. And a useful thing about this system is, as I mentioned, we already have a pretty good idea of the major loci underlying floral color, or I'm measuring it as floral hue here, but essentially it's color. And here I've got the Sierras and Crater Lake stuff. And as we see in both of these populations, there's a nice big outlier, I guess, centered on F3'5'H, which, as I mentioned before, is a known major floral color locus in penstemon and lots of other floral or plants. But interestingly, there's also a peak in the Sierras at F3'H, which is another anthocyanin biosynthesis enzyme, which isn't present at Crater Lake, suggesting that maybe there's like an additional sort of locus that's, I guess, important for the color in the Sierras. But as we were interested in stuff other than color because we kind of already knew what this would look like. I'm not going to like walk through all of our results here because, as I mentioned, this is sort of still a work in progress. But what I can tell you is that most of our traits do appear to be polygenic, some more than others. And we do have lots of potentially interesting candidate genes. So I picked here floral tube length at Crater Lake. And so there are some interesting things that are probably happening. Need to sort of drill down into some of these results and give you some clear pictures of this at a later date. All right, so last, this question of selection. Is pollination syndrome actually an effective mechanism for reproductive isolation? You can think of this as sort of this hypothesis where essentially if floral traits confer strong reproductive isolation, we would expect that loci that actually underlie that variation should not be moving between species. And just as sort of like a way to look at this, I use this approach called genomic clines. And I'm not going to get into the details of that analysis, but essentially you can identify loci that are consistent with selection against integration. So essentially stuff that doesn't appear to be moving into different parent backgrounds, essentially. And really interestingly, and these are called steep clines. Really interestingly, over 90% of all of the steep clines that we're finding are on this single chromosome, which I've labeled 1086. And all of those essentially are centered within 10 megabase pairs on this F3'5'H locus. And so while this doesn't necessarily suggest that F3'5'H is like the source of this strong selection, I would argue that it does seem to be something really interesting happening on this chromosome, and the major floral color locus is right at the center of that. So more to come on this as well, but just some food for thought, I suppose. So just a quick summary here. Our hybrid zones have a mix of early generation and multi-generational back crosses. Our GWAS seems to be working pretty well, but still in progress. And maybe there's an additional floral color locus that we should be thinking about. And our signatures of selection against integration are sort of all clustering together, and is consistent with this idea that maybe pollinator specialization is actually a pretty effective mechanism for reproductive isolation. With that, I'll thank my lab and funding sources, and just give a shameless plug that I am starting my lab at Mississippi State next fall, and I am recruiting grad students to start as early as autumn of 2025. So if you're interested or know someone, definitely get in touch. All right, thanks. Oh, yeah.
Did you roll out an inversion in that 5 megabase region?
No, I have not rolled out inversion at all. That's something I'm working on. It's weird. We've done some synteny plots between other genomes, and that seems to be hard to assemble, and is not synthetic with the other genome. So there's something weird happening, but not sure what at the moment.